welcome to my channel. Today we'll be talking about carbon dioxide fixation. So basically what that means is that carbon dioxide will be fixed to become a sugar. In this case it would be a derivative of glucose, which is fructose. Again, these are two sugars. So there are a specific type of organism that can do this kind of mechanism. If you put into perspective what eukaryotes are capable of doing, eukaryotes respire oxygen and create carbon dioxide. Never in any mechanism do we take in carbon dioxide and fixate that. So that excludes eukaryotes, animals, and all of that kingdom basically from being able to fixate carbon dioxide. That leaves us with prokaryotes, therefore bacteria and microbes. Specifically, autotrophs are the ones responsible for doing this type of fixation. So autotrophs are able to fixate carbon dioxide, which is an inorganic molecule for them, and are able to create a carbon source to create a precursor metabolite. So what that means is that an autotroph fixates a carbon dioxide molecule to be able to extract a carbon from it as fuel to create then a precursor metabolite. Precursor metabolites are 13 specific compounds that are used to create building blocks. So these precursor metabolites create building blocks, which building blocks create something else. So we need first the down to the very detail of compounds to create a building block to then build up and make a house, basically. So you need the precursor metabolites. The building blocks themselves are molecules that are synthesized by these precursor metabolites, and they are then polymerized to build cellular macromolecules. Macromolecules can be amino acids that are then used to create proteins. So amino acids are what's used to create proteins. So just to reiterate all that, we have CO2 fixation, which means that CO2 is being fixated to create a fructose molecule, which is a sugar. The only organisms that are able to do that are autotrophs. So how does it look down to the molecular level? Here behind me I have the skeletal structures of the molecules involved in the fixation. So we have carbon dioxide, as we know, and that would be denoted as carbon, and dioxide means two oxygens. So it has two double bonded oxygens on that carbon, and this is what the skeletal structure of carbon dioxide looks like. That is what's going to be added to a molecule. There is going to be a molecule that accepts the carbon dioxide in this fixation. So that molecule itself would be ribulose 1,5-biphosphate. It has 5 carbons here, and it says biphosphate, which means it has 2 phosphates. 1,5 denotes the actual carbon on that molecule that the phosphate is attached to. So we have the 1 carbon, 2, 3, 4, 5. So 1 and 5 both have the phosphates attached. So the carbon dioxide will attach to this ribulose biphosphate. The way that it will attach to that molecule is that this carbon will attach to one of the carbons on the ribulose biphosphate. The specific one that it has to bind to would be this double bonded O. The reason being is that the next proton over can be kicked off, and these electrons move down to this molecule here to create a double bond instead of a single bond, and this becomes a single bond because we're adding now a new carbon. The actual attaching of the skeletal structures, that's organic chemistry, but this is helpful in knowing what the actual reaction looks like to the chemical level. So the carbon will come down here, make all those movements, and since it's double bonded on both sides, it also needs to move some electrons out to this oxygen. So one of them will become a single bond, and this will become a double bond. This is what the intermediate looks like, which is the carbon dioxide actually bonded to the 5-carbon ribulose. So if you notice, now we have 1, 2, 3 carbons here, and we have 1, 2, 3 down here. We can now split this into 2 even molecules where they each have 3 carbons, and they each will have their own phosphate. So when you split that, you get 2 molecules of 3-phosphoglycerate, which means that the 3 is a position again where the phosphate is attached on that molecule, specifically the carbon number. So carbon 1, 2, 3, the 3rd carbon. The numbering will make sense again if you've taken organic chemistry or you plan to. The numbering here is denoted by a specific rule as to why this one is 1 and that one is 3. But for now, just know that the side that has H2O has the phosphate attached on both of the molecules. So that's the very first step that you will see in carbon dioxide fixation, which is actually having the carbon dioxide attached to a molecule. And just to emphasize everything, Rubisco is the one responsible for allowing the carbon dioxide to even bind to this molecule. Without Rubisco, this enzyme, we wouldn't be able to have this addition. So we must have these 3 factors in order to have this intermediate happen in the first place. So Rubisco is ribulose biphosphate carboxylase. So here we have the actual Calvin cycle. This is what the cycle looks like with all the molecules involved, everything as far as energy added and all that good stuff. So it looks like there's a lot going on, but once you break down the concepts, it'll make sense, and you can just go through this in an overview type of way. So we first mentioned that we have CO2s that are added. To this reaction, we must have a specific number of carbon dioxides added to get a molecule. So we need to add specifically 6 molecules of CO2. And again, that's facilitated through Rubisco. We did see previously that CO2 binds to the 3-phosphoglycerate, which is this molecule here, facilitated by this enzyme. So this is added to the ribulose biphosphate, the ribulose 1,5-biphosphate, which has the 2 phosphates here. So this CO2 will bind to this through the catalysis of Rubisco, and you end up with 12 molecules of the 3-phosphoglycerate. 3 because position 3 has the phosphate. That's the very first step we saw at a molecular level. So that's the very first step, and just keep that in mind. So next up, what we can break down is the actual cycle that has 3 quadrants in it. The first quadrant would be carbon fixation, which right now we just went over. The second part of this quadrant would be reduction and sugar production. And finally, regeneration of RUBP, which is ribulose biphosphate. So after this fixation, now we can go into the reduction and sugar production. Once we have those 12 molecules that were split, once we have those 12 molecules, we need to add 12 molecules of ATP. We need to introduce an energy source. So we added these ATPs, and ATP will allow you to phosphorylate your molecule. So it'll give one of its phosphates, and now this phosphoglycerate becomes biphosphoglycerate. So IP is denoted to be biphosphate, so it's 2 phosphates now. 3 carbons still, but now it's on the not only the 3 position, but the 1 position. So now we need to reduce this. What we can reduce with is a reducing power molecule. So a reducing power molecule would be NADPH. This molecule will reduce the power of the compound. We need to add 12 of those molecules as well. We introduced 12 ATPs, so we introduce 12 NADPH. That reduces the molecule to now have only one phosphate, where it previously had two before being reduced, now has one. In being reduced, it would also produce a sugar. So we have now a biphosphate that became a 1-phosphate molecule, now called glyceraldehyde, 3P. So on the 3 carbon we have the phosphate. It now becomes 12 of these glyceraldehydes that get broken down into 1 fructose and then 10 glyceraldehydes. Two of these glyceraldehydes were used and 1 fructose came out here, and now we have glyceraldehyde. We can think of this whole thing as an analogy with a car, and the car would be the ribulose biphosphate. Carbon dioxide is the passenger. So the car will always pick up this CO2, go through a couple stops here, and finally drop off a sugar. Go ahead, and that's where the carbon, this is where the carbon becomes a sugar, drops it off, car is empty, and is ready for another load of CO2. So just to reiterate, we have fixated our carbon dioxide, we have reduced it and produced a sugar out of it by adding an energy source and adding a reducing power molecule. We get a sugar molecule. Now we're finally here, we are regenerating the ribulose biphosphate. So now these 10 glyceraldehydes will become, from a 3-carbon molecule, will become a 5-carbon molecule with the phosphate attached to the 5th carbon. It went from 10 molecules to now 6 of these ribuloses. We now add another source of ATP. We had 6 ATPs here, and the ATP will give one of its phosphates away. ATP is triphosphate, so it has 3 phosphates. It gives one away, so now this molecule went from having 1 phosphate to having 2 phosphates. Now it's in the format it must be to be able to pick up another CO2. That's where you know life finally regenerated. That's basically carbon dioxide fixation in a nutshell. This is the whole cycle. It wasn't as bad as it seemed. I hope not. Okay, so we went over the precursor metabolites. We went over that autotrophs are the only ones that can fixate carbon dioxide to create sugar, and we really then went over the cycle of what the molecules go through and create what, where, and what's added. So now finally we can put that down in an equation and see what the reaction equation is to this fixation. What are the reactants, what are we introducing, and what are the products? What are we creating from these reactants? The reactants that we started with are 6 CO2 molecules. We also had to add the reducing power, which was 12 NADPH molecules. We also added a total of 18 ATP molecules. We added 12 in the beginning to add that phosphate and create the biphosphoglycerate. And at the end we added 6 ATPs to finally regenerate that ribulose biphosphate to be now a 1,5 ribulose biphosphate. So these are our reactants. Those will make our products, which are C6H12O6, which is sugar. This is always denoted to be sugar, C6H12O6. And in parentheses we have a phosphate group attached. So we have that fructose with the 6P, which means the phosphate is attached to the 6th carbon of that sugar. We have also in the products 12 NADP plus molecules. these look similar to NADP, look similar to NADPH. The difference here is that this, it's the same molecule, but this one got reduced. So it lost the proton because it reduced something, and it became NADP+. Now we also have, in the products, we have 18 ADP. Similar again to ATP, ATP has a triphosphate. It gave one away both times that it was introduced, so it ends up being a diphosphate where it has two phosphates. So in the products we'll have 18 ADPs. And finally we have 17 Pi, which means phosphorus ions. So they're just floating around. So this is the reaction equation to carbon fixation. Let me know if you have any questions, and thank you.